Good afternoon. I hope my voice is audible. Very good afternoon to the student who have joined. Please let me know if you can hear my voice and also let me know if the presentation is visible to you. Okay, thank you for confirming. We will be starting our session at 4 p.m. So kindly wait till then. Good afternoon to the students. We will be starting our session from 4 p.m. Till then, please wait. I hope my voice is audible and you can see the presentation. I mean, you can see my screen clearly. If not, please let me know. So today it is our last session. If you have any doubt or any query after today's class, you can let me know via email. Okay, I think I have shared my email ID earlier, so I will share it again at the end of today's session. So if you have any doubt or any query after today's class, please feel free to let me know via email. Okay, so it's 4 p.m. I think we should start our session. The session is open, so meanwhile, other students can join accordingly. So, very good afternoon to the students who have joined. My name is Mousumi Deb, and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL SWAYAM course of Medicinal Chemistry, and today it is our last class. It is a summary session where I will summarize a few topics of your course. So basically, in our course of medicinal chemistry, what we have done is now we have discussed various aspects of the subject. Okay, right? We have started from the very the enzymes and the protein, and then we followed by the enzyme catalysis and inhibition. Then we discussed about the pharmacokinetics, pharmacodynamics, metabolism, drug administration, everything we have discussed, followed by the therapeutics. Okay, so today it will be the last regular class. Okay, after that there won't be any class, so after that, after today's class, if you have any doubt or any query before your examination, you can let your queries know via the email ID that I will share at the end. Okay, and otherwise, I think whatever your course is, I mean the topics that are in your course, I think if you follow the book and the material whatever has been taught in the problem solving sessions, I think that will be sufficient for you for your examination purpose. Okay. Okay. So today it is a summary session. I hope my voice is audible and you can see the presentation clearly. If not, you can let me know. Okay. So today what we will do is I will also today, I mean, we will discuss similarly the MCQ questions. Okay. So I will share 20 MCQ question, and all questions are from different weekly courses from your course. Okay. So it's not like the particular question is from a particular week. No, but all the questions are from each and every, I mean, here and there. So I have collected few questions. So, and few topics that I have not discussed earlier. So I thought to, I mean, discuss it in today's class. And also, since there was a doubt regarding the antibacterial agents and antibiotics, how to remember this vast, I mean, list of therapeutics are there as an antibacterial agent. So, at the end of today's session, I will discuss that particular thing also very briefly. I have, I mean, discuss that. I will discuss that antibiotic part so that it will be easier for you to remember. So, I will discuss it in that way. Okay. All the materials are already uploaded. Whatever I have taught till now, so that I have already uploaded on the SWAYAM portal. You have to just go to the medicinal chemistry course and click on the problem solving session recording. On clicking on this particular, I mean, link, you will be redirected to a page which looks like this. This has all the YouTube links of the class recordings. Okay, all the you can see all the YouTube links are there, and each and every class whatever we have done. And this is the link for a Google Drive. This Google Drive contain all the presentations. Okay, whatever slides I have taught till now. Now, all the 12 slides are there, and today's slides also I will upload it after today's class. Okay, so you can go to this drive, you can download the material, and you can read. Okay. So let me start today's session. Since today is the last class, I will hope that all of you will try to answer the questions. Okay, now because it is the last class and now you should be able to answer the questions. Okay, even if your answer is wrong, it's fine. But since we are at the end, so I will expect that you will at least answer few questions correctly. Okay. So now let me go to the first question of today's session. So the first question says, which of the following drug is withdrawn from the market due to its damaging effect on liver cells? Your options are number A, Practolol, number B, Tylenolic acid, number C, Cerivastatin, and number D, phenylbutazone. Which of the following drug is withdrawn from the market due to its damaging effect on the liver cells? Practolol, Tylenolic acid, Cerivastatin, phenylbutazone. After showing every question, I will wait for 2 minutes, then I will check the chat box, okay? And then we will discuss that particular topic in detail. It's similar to what we do in our everyday class, okay? Which of the following drug is withdrawn from the market due to its damaging effect on liver cells? Practolol, Tylenolic acid, Cerivastatin, or phenylbutazone? Which of the following drug is withdrawn from the market due to its damaging effect on liver cells? Practolol, Tylenolic acid, Cerivastatin, or phenylbutazone? Try to answer the question. I will check the chat box in a minute. Okay. Okay, let me check the chat box now. Okay. Two answers I have received, and both are saying that number C is the correct one. Okay, let me now check if number C is the correct answer or is there any other answer which is the correct one. The correct answer is it is number B. Tylenolic acid is the drug, okay, that is withdrawn from the market due to its damaging effect on the liver cells. So this is the structure of the tylenolic acid. So this actually comes to the phase 4 of clinical trials. So when we have learned about the clinical trials, first is the normal drug discovery process that has the all the pharmacokinetic, pharmacodynamic study, the synthesis, heat to lead optimization, followed by all the preclinical studies on the animal model and these things. After that, we know that the compound or drug is, it goes to the clinical trial, which is basically the human trials. Okay, the clinical trials, it first starts with a few number of people, then it generally it will proceed with a larger number of patients, right? And this phase 1, phase 2, and phase 3 are the clinical trials, and after that the drug is approved. The drug is first is it is monitored by FDA, which is the Food and Drug Administration of okay. So that is an USA based agency, governing body, that tests the drug and then it is released to the market, okay, for human trials. But after a drug is it goes to the human it is goes to market for using the larger number of population. Some drug, they may show some kind of damaging effect to our body, like they may develop some resistance, or they may show you some harmful effect. Because see, whenever clinical trials are done, they are done on a thousand of number of people, right? 1,000 or 10,000 or 1 lakh or so on. But when a drug is going to market, it will be taken by billions of people, right? So in those people, this drug can give rise to some kind of damaging effect or some side effect or some genetic disorder or everything can be drawn. So there is a thing which is known as the post-market surveillance. So this that is also a part of a clinical trial. Okay, so after some time, the drug is in market. After some time, it is monitored to check if the drug is giving rise to some unexpected or rare side effect. Okay, that is the phase 4 clinical studies. Tylenic acid is one of those drugs which were withdrawn from the market because of its side effects. Okay, so the drug is so after a drug is in placed in a particular market, now it can be prescribed, but still monitored for the effectiveness and for any rare or unexpected effects. In a sense, this phase never-ending process, and as unexpected side effects may crop up many years after introduction of the drug. Okay, in the UK, the Medicines Committee ran a voluntary yellow card scheme where doctors and pharmacists report suspected adverse reaction for the drug. This system has revealed serious side effects for a number of drugs that had been put to the market. For example, beta blocker practical had to be withdrawn after several years to use because some patients suffered from blindness and even death. The toxic side effects were unpredictable and are still not understood, and so it has not been possible to develop the test for this effect. The diuretic agent tylenic acid had to be withdrawn from the market because it damaged liver cells 1 out of every 10,000 patients. The anti-inflammatory agent phenylbutazone can cause a rare but fatal side effect in 22 patients out of every million treated with the drug. Now, such a rare toxic effect would clearly not be detected during the phase 3 clinical trial. A more recent example is the serivastatin, which was marketed as a potent anti-cholesterol drug. Unfortunately, it has to be withdrawn in 2001 as a result of the adverse drug-drug interactions, which resulted in muscle damage and deaths of the 40 people worldwide. Okay. So this is the so all the drugs that are mentioned in this question, practical, tylenic acid, serivastatin, phenylbutazone, all had some sort of side effects, and all the side effects are detected during the phase 4 clinical trials. Okay, and so it is important for you to remember the names of the drug which were withdrawn from the market due to this kind of I mean side effect. That is the practical because of their because they patients suffer from blindness and death because of the use of this drug. Okay, tylenic acid, as I have mentioned in this question, it give rise to the liver damage. Okay, and then in this phenylbutazone, which can be cause a rare or fatal side effect. Okay, and serivastatin, which also have some drug-drug interaction, which is adverse in nature, and because of that, all these drugs have to be withdrawn from the market. Okay. So this is the importance of the phase 4 clinical trials, because after phase 3, although the drug is approved for I mean market, some side effects can always generate, and when the drug goes to the higher population of the patient. Okay. So number B was the correct answer. Now let me go to the next problem. Problem number 2 says, which of the following statements regarding lipophilic efficiency is false? Your options are number A, optimizing LIPE, or this LIPE means this lipophilic efficiency. I have already mentioned you in medicinal chemistry, we actually use various shorthand notations and acronyms for various topics, okay, or various things. So it is important for you to identify those and to remember, okay. So because not every time the question will be given to you with the I mean full word of so the acronyms or the shorthand notations can be given to you in each and every in questions. Okay. So which of the following statement regarding lipophilic efficiency is false? Your options are number A. Optimizing lipophilic LIP involves parallel optimization of potency. Number B, drugs with a good level of lipophilic efficiency will have a high hydrophobic character. Number C, drugs with a good level of lipophilic efficiency will have a low hydrophobic character. And number D, lipophilic efficiency can be calculated by the equation LIEP equal to PIC50 minus C log D. Which of the following statement regarding the lipophilic efficiency is false? Read the options very carefully and then let me know, okay? I will check the chat box after a minute or two. Then I will discuss what is the lipophilic efficiency and what is the importance of this. Again, I will repeat the options. Number A, optimizing lipophilic efficiency involves a parallel optimization of potency. Number B, drugs with a good level of lipophilic efficiency will have a high hydrophobic character. Number C, drugs with a good level of lipophilic efficiency will have a low hydrophobic character. Number D, lipophilic efficiency can be calculated by the equation LIPE equal to PIC50 minus C log D. Which of the following statements regarding lipophilic efficiency is false? Let me check the chat box. Okay, number D, number C, and number B. So I have received three different options as the answer. Okay, that's good. Let me now check which of the following will be the correct answer. Okay, so yeah. So the correct answer will be it is number B. Drugs with a good level of lipophilic efficiency will have a high hydrophobic character. This statement is false. Okay. Now let us discuss about the testing procedures and the lipophilic efficiency, how it depends on the testing procedure depends on it. So when investigating the structure activity relationships or SAR for drug target binding interaction, biological testing should involve in vitro test, for example, inhibition studies on isolated enzyme or binding studies on the membrane bound receptors in a whole cells. The results then show conclusively which binding groups are important for in the drug-drug interaction. If in vivo testing is carried out, the results are less clear-cut because loss of activity may be due to the inability of the drug to reach its target rather than the reduced drug-target interaction. However, in vivo testing may reveal functional groups that are important in protecting or assisting the drug it's passing through the body that would not be revealed by the in vitro testing. So there are two type of testing we have so not two, there are three type of testing actually. First is the in silico, which is the computer-based techniques that are the computational techniques. Okay. Then we have this in vitro studies. In vitro studies are done on the isolated enzymes. For example, you want to develop a enzyme inhibitor. So in vitro study will be to check if the compound is inhibiting that particular enzyme, but it will be on glass, not in the body of the particular target. Okay. And then we have the in vivo study when the drug is pushed to an animal or, for example, yes, the drug is pushed into an animal like mice or rabbit or this kind of animal, and then we have to check if the particular drug is hitting the target or not. So each and every test have their own benefits. For example, in vitro tests are good because this test show actually how the drug target interaction happens. But in vitro, in vivo tests are now when a drug is given to the body, there are various factors that are come into play, right? Because we have to check if the drug is reaching to that particular target, its pharmacokinetic and pharmacodynamic stability, and so on. The advantages of in vivo study is to check that if the drug is so which functional groups are vulnerable in the particular drug which need to be protected in form of a pro drug or not. So these things are reported. These things cannot be revealed by the in vitro testing, right? So NMR spectroscopy can be also used as a testing procedure as mentioned. So it is a little the designing a drug has an optimum interaction with the target if the undesirable pharmacokinetic properties. Okay, so calculating a structures by hydrophobicity can provide an indication as to whether it is likely to suffer from pharmacokinetic problem. This is because hydrophobic drugs have found to be more prone to adverse pharmacokinetic properties. Okay, for example, they are more likely to interact with the other protein target, resulting in unwanted side effects. They are generally less soluble, show poor permeability, and are more likely to produce toxic metabolites. The hydrophobic nature of a drug can be calculated by C log D value. In recent years, several research groups have optimized the drugs by optimizing the lipophilic efficiency of the drug. Okay, so lipophilic efficiency where this is equal to PIK or PIC 50 minus C log D. Okay, so this is the formula of the lipophilic efficiency. The drugs with a good level of lipophilic efficiency will have a good activity, okay, and low hydrophobic character. This is important to remember. Lipophilic efficiency, sometimes when we are, hearing the term lipophilic efficiency, it may seem that, okay, it is synonymous with a drug to be more hydrophobic and which is not actually. So because when a drug has a good lipophilic efficiency, it will mean that it has a low hydrophobic character and good efficiency or good activity, okay. The optimizing lipophilic efficiency involves in parallel optimization of potency and hydrophobicity. This quantitative method of optimizing both in the pharmacodynamic and pharmacokinetic has been called the property-based drug design. We know what is the structure-based drug design and what is the fragment-based drug design. So there is another thing which is the property-based drug design. Okay, what is this? This is the method of optimizing both the pharmacokinetic and pharmacodynamic properties of a drug. This and it was used in the structure-based study of design of the drug crizotinib. Okay, so this is an example which is developed with this method of the property-based drug design. Okay, so remember what is lipophilic efficiency, what is the formula of lipophilic efficiency, what is the important parts of the lipophilic efficiency. Okay, so these things you have to remember and understand. Okay, so number B was the correct answer. Now I will go to the next problem. Problem number 3 says smallpox and yellow fever are serious viral infections, but this can be tackled by your options are number A antiviral drugs, number B natural product, number C vaccination, and number D immunotherapy. Smallpox and yellow fever are serious viral infections, but these can be tackled by which of the following? Antiviral drugs, natural product, vaccination, or immunotherapy. I will check your answer after a minute, okay? Let me know A, B, C, or D, which of the following will be the correct answer. Smallpox and yellow fever are serious viral infections, but these can be tackled by which of the following? Antiviral drugs, natural product, vaccination, or immunotherapy. Let me know A, B, C, or D, which of the following will be the correct answer. Let me check the chat box. Okay, number C, number D, and number A. So I think I have again received three different answer A, D, and C. Okay, let me check which of the following is the correct answer for our problem number 3. So the correct answer is number C, vaccination. Okay, so smallpox and yellow fever, these viral infections can be tackled by the vaccination process. So one of the ways by which we can protect ourselves from the viral infection is to use vaccination, and the number of diseases that we have looked as far as polio, measles, and mumps are actually mainly targeted by vaccination. So smallpox and yellow fever are also sort of the serious disease which can be countered by vaccination. So what is vaccination? So vaccination works by introducing the body to foreign material which bears molecular similarity to some component of the virus, but which lacks its infectious nature or toxic effects. The body is then has the opportunity to recognize the molecular fingerprint of the virus, for example, specific antigens of that virus, and the immune system is primed to attack the virus. Should in attack the virus should infect the body. When it will infect the body, that usually a killed or weakened form of the virus is administered, so that does not lead to any infection itself. Alternatively, fragments of the virus or subunit vaccine can be used if they display characteristic antigen. Vaccination is a preventive approach and is not usually effective on the patient who have already become infected. Okay, so you have to give a patient particular vaccination before he or she has encountered that disease. Okay, for if somebody has gotten this, smallpox, or they have gotten some yellow fever or this kind of disease, after that, if you give him or her the vaccination, it won't work. You have to give it earlier. So basically, how this vaccination thing works. So first is this, your inactivated vaccine based on the chemically, and so first this is the so this is the structure of a virus, right? So what is the structure of virus? We have already discussed in our I think 11th class. We have discussed about the antiviral drugs. There I have discussed about the structure of the virus and various parts, like it has a membrane, it has an envelope protein, then we have a spike protein, genome of the RNA nucleocapsid protein. So all these are the proteins. So this is the spike protein of the virus which can be used as this particular vaccine, or inactivated vaccine based on the chemically or physically inactivated virus grown in the cell culture. Live attenuated vaccine utilizes a weakened version of limited replication or extent of the living virus, where vector vaccine adapts the existing successful or safe modified viral vectors to express the coronavirus protein. DNA vaccine contains the circular DNA that encodes the protein of the interest. RNA vaccine uses mRNA packed with a lipid nanoparticle. Then we have the recombinant subunit vaccine composed of the synthetic nanoparticle coated with a purified viral antigen. And virus-like particles vaccine contain the multiprotein structures that mimic the virus but yet divert of the viral genome. Okay, so these are the techniques by which the vaccination can be done. Each and every there are for different disease we have different types of viruses. Okay, so these are the basics of the vaccination. So I will go to the next problem then. Problem number 4 says, which of the following is a nitrosourea alkylating agent? Your options are number A, lomustine, number B, carmustine, number C, streptozotocin, and number D, all of the above. Which of the following is a nitrosourea alkylating agent? Lomustine, carmustine, streptozotocin, or all of the above? Let me know which of the following will be the correct answer for our fourth problem. Which of the following is a nitrosourea alkylating agent? Lomustine, carmustine, streptozotocin, or all of the above? I will wait again for a minute, then I will check the chat box. Which of the following is a nitrosourea alkylating agent? Lomustine, carmustine, streptozotocin, or all of the above? Which of the following is a nitrosourea alkylating agent? Let me check the chat box. Let's see if I have received any answer for the question number 4. Okay, number D, number D. So I have received three options and all are saying that number D is the correct one. Okay, very good. Let me now check the answers. The correct answer will be yes, number D. All of these are the nitrosourea alkylating agents. So I think I have discussed about the nitrosourea alkylating agents or alkylating agents. Okay. So nitrosoureas, the anti-cancer agents, lomustine and carmustine. So this is the structure of lomustine. This is the structure of carmustine, and this is the structure of the streptozotocin. Okay. These are the alkylating agents, right? We have learned about the alkylating agents when I was discussing about the nucleic acid as a drug target. I think it is in our in your sixth week or fifth week, I don't remember exactly. Hopefully, I think it is the sixth week course content. Okay, so the anti-cancer drugs, lomustine and carmustine, were discovered in 1960s and chloroethyl nitrosoureas, which decompose spontaneously in the body to form two active compounds, an alkylating agent and an carbomylating agent. Okay. Mm, so the organic isocyanate, which is formed that carbomolates the lysine residue in the proteins and may inactivate DNA repair enzyme. The alkylating agent acts initially with a guanine moiety with one strand of DNA and then the guanine and cytosine unit to the other strand to produce the intrastrand cross-linking. Streptozotocin is a naturally occurring nitrosourea isolated from the Streptomyces acarinomys. So this is the structure of your normally lomustine and carmustine both have this kind of structure. So this is a nitrosourea kind of group. It forms isocyanate and also it forms an alkylating agent. This is isocyanate. It actually carbomylates. Carbomylation happen means this introduces this CONHR group to a particular protein residue that has a lysine. Okay, means it forms this kind of urea group in case of I mean amide linkage introduces to the lysine residue, and also we have this kind of alkylating agent which is formed, which causes the DNA alkylation with the cysteine and your guanine. So this is the structure of cysteine and this is the structure of guanine. It is important for you to remember the structures of the all nucleobases like adenine, guanine, thymine, cytosine, and uracil. Adenine and guanine, they are little big structure because these are the purine structures, but the pyrimidines are, I think, they are easier to remember. The cytosine, uracil, and thymine. Okay, so in case of the guanine, there are 1, 2, 3, 4, 5, 6, 7, 8, 9 points are there. Okay, and this is so numbering is important. How the numbering is happening, it is very important. And so here, this number is on sixth position. This O, this carbonyl group is alkylation here occurs with the nitrosourea, and in case of the third nitrogen of cytosine, the alkylation happens with the nitrosourea. Okay, and remember, in case of the N1, the DNA is binds with the cytosine. There is a DNA binding, and in case of the guanine, in on the N9, the DNA binding happens. Okay, so this is also important. The alkylation. The alkylation sites on the guanine and cytosine for in case of nitrosourea, we have to remember. So here, as you can see, here it happens with in case of the guanine, and here it happens in case of the cytosine. Okay. On in for more details of the alkylating agent, already I have discussed lots of regarding this alkylating agents in your particular nucleic acid as a drug target, that chapter. You can go through that particular material. Okay, you will find more examples of the alkylating agents there. Now I will go to the fifth question. It says which of the following is not a fourth generation cephalosporin? Your option as are number A, cephalexin, number B, cefoxitin, number C, ceftazidime, and number D, cepiromen. Which of the following is not a fourth generation cephalosporin? Cephalexin, cefoxitin, ceftazidime, or cepirome? A, B, C, or D. Now, your options are number A and B, number B, A, B, and D, number C, A, B, and C, and number D, C, and D. Which of the following is not a fourth generation cephalosporin? Cephalexin, cefoxitin, ceftazidime, or cefpirone? Number A or B. Number B is A, B, and D, and number C is the A, B, and C, and number D is C and D. I will check your answers after a minute. Try to answer the question. Let me check the chat box. Let's see if there is any answer for our problem number 5. Okay, number C, number A, number A. Okay, I think number A and number C are the two options that I have received for this question. Hmm. Okay. Others are also are not answering. I want all of you to answer, okay? Because today is the last class, so I want all of you to participate and answer. So the correct answer will be, it is number C, A, B, and C. Okay, A, B, and C. Now, this will be, these are not the fourth generation cephalosporin. Okay, so this is the structure of your Cephirome, which is a fourth generation, cephalosporin. So the clinical aspect of cephalosporins. So there are lots of details are given here. You do not need to memorize all those, but try to remember the names of the cephalosporin of the each and every generation. Okay. So in general, cephalosporins are useful broad spectrum antibacterial agent for the treatment of the septicemia, pneumonia, meningitis, binary tract infection, peritonitis, and urinary tract infection. Cephalosporin C itself has been used in the treatment of the urinary tract infection, and it is found to concentrate on in the urine and survive in body's hydrolytic enzymes. So the first generation cephalosporin have good activity against the gram-positive cocci, and it can be used to treat some community-derived gram-negative infection. The infection are not caught in the hospital. Okay, they can be also used against S. aureus and streptococcal infections when the penicillins have been avoided. Cephalethine is more active than the penicillin G against some gram-negative bacteria and is less likely to cause allergic reaction. It can also be used against the beta-lactamase producing the S. aureus strain. Now, cephalexin is useful in treatment of urinary tract infection which do not respond to other drugs or which occur in pregnancy. It is also useful in treating the infections of the respiratory tract, ear, skin, and mouth. Cefaxolin is recommended for use to the prophylactic to prevent the infection when surgical procedures are used to implant foreign bodies. Okay. So these three are the first generation cephalosporin. Now, second generation cephalosporin in general, second generation cephalosporin have the variable activity against the gram-positive bacteria, but increased activity against the gram-negative one. Cefoxitin is the active against the bowel flora, including the this Bacteroides fragilis, and was recommended for the peritonitis. The cefoxim was a wide spectrum activity and is useful against the organisms which have become resistant to the penicillin. However, it is not active against the difficult bacteria such as the P. aeruginosa, and it is used to clinically against the Neisseria gonorrhoeae and the respiratory infection by the influenza virus or other susceptible strains of the pneumonia. Okay, so it is also used for the surgical prophylaxis as well as the treatment of the Lyme disease. Cefoxitin is used in the surgical prophylaxis and for the treatment of the gonorrhea, meningitis, and infections caused by the Haemophilus epiglottis. Now, third generation cephalosporins have good activity against the gram-negative bacteria, but they vary with their activity against the gram-positive one. The ability to attack the P. aeruginosa also varies from the structure, and they have lacked the air activity against the MRSA organisms. Okay. And the Enterobacteria species. Ceftazidime is an injectable cephalosporin which has excellent activity against the P. aeruginosa as well as the gram-negative bacteria. Now, because the drug can cross the blood-brain barrier, it can be used to treat the meningitis. It is very important. Okay, let me highlight it. Cefaxetine is an injectable cephalosporin that has a excellent inactivity as well as this drug can cross the blood-brain barrier, so it can be used for the meningitis. This is a disease for the brain. Okay, the when the bacterial infection happens in the brain, in that case, this drug can be used because it can cross the blood-brain barrier. Compared to the other aminothiazole structure, this has a good activity against Streptococci, but loses its activity against the strains of the methyl susceptible S. aureus. This is because the disease binding of the affinity for the transpeptidase enzyme is present in the S. aureus. Ceftriaxone is used in the surgical prophylaxis. Okay. And fourth generation and fifth generation one, the fourth generation cephalosporin has the activity against the gram-positive one and broad variety of bacteria species. Cefalopyrome is administered as the intravenous injection or infusion and has been against using various of the sensitive gram-positive and gram-negative bacteria. Cephalosporin formamide has been licensed for the treatment of the bacterial pneumonia and acute skin infection. So as I have mentioned, you do not need to memorize all the details, okay? But try to remember which drug is from the which generation, and some exceptional drugs like, for example, this ceftazidime, which can be used in treatment of meningitis because it has the ability to cross the blood-brain barrier. These things try to remember, okay? Now I will go to the next problem. Problem number six says, which of the drugs can be used for the treatment of CMV retinosis? We check once. I need to understand the generations. Okay, so the generation actually, what happens whenever a drug is first developed, right? So it is known as the your first generation. The more we modify the particular drug and we keep modifying the particular drug, and the new generation comes. Okay, for example, so there is no way that you can understand the generation. So basically, when a series of drugs are developed as a first generation drug, there may be some kind of side effects, or the when the back, for example, the antibacterial ones, let's say. Okay, so the drugs, they, when first generation drugs are administered, sometimes after that the bacteria may develop the resistance against the particular drugs, and it is the same for any kind of antiviral, anti-cancer, and so on. So when the resistance happen, we need to change the drugs. We need to vary that particular generation a little bit to develop another generation or another series, which is known as the second generation, and it happens after some time. Okay, so that's how the generation these things are coming. So there is no way that you can understand that generation, because it is a very vast thing, and it is different from different diseases and different series of the therapeutics. Okay. Okay, now I will be going to the sixth question. It says that which of the following drugs is used for the CMV retinosis? Your options are number A, desiclovir, number B, valacyclovir, number C, trifluridine, and number D, foscarnet. Which of the following drugs is used for the treatment of the CMV retinosis? Desiclovir, valacyclovir, trifluridine, or foscarnet? Which of the following drugs is used for the treatment of CMV retinosis? I will wait for a minute, then I will check the chat box, okay? Let me know A, B, C, or D, which of the following will be the correct answer. Which of the following drugs is used for the treatment of CMV retinosis? Desiclovir, valacyclovir, trifluridine, or foscarnet? Which of the following drugs is used for the treatment of CMV retinosis? Let me check the chat box now. Okay, number B, number C, and number D. So I have received three different answers. Okay, let me check which of the following is the correct answer. So the correct answer will be number D. Foscarnet is the drug which is used for the treatment of the CMV retinosis. So these are the example of the viral DNA polymerase inhibitors. Okay. So these are the various clinical aspects of the DNA polymerase inhibitors. Again, I am saying you do not need to memorize all these things, but a few points that you need to be that you need to remember. Okay, so acyclovir is represented as a revolution in the treatment of the herpes infection, being the first relatively safe and non-toxic drug to be used systematically. So the first one is the acyclovir. Okay, so it is used for the treatment of the infection due to the herpes simplex virus 1 and 2. For example, this herpes simplex encephalitis and the genital herpes, as well as the VZV or chickenpox or the shingles. Unfortunately, strains of the herpes are appearing which are resistant to acyclovir. This can rise as a result of the mutation and either for the viral thymidine kinase enzyme such as no longer phosphorylates acyclovir or viral DNA polymerase such that it no longer recognizes the activated drug. Acyclovir is not effective against all type of herpes virus, and there are 8 herpes virus that are divided into 3 subfamilies. Acyclovir is effective against the alpha subfamily, not the beta subfamily, because the latter produces the different thymidine kinase and fails to phosphorylate the drug. This is important. Acyclovir, how it is it has a pro-drug, so it is a so how it gets activated. So all we have discussed in our that class, I think it is the 11th class when we were discussing about the antiviral drugs. You can go through the material. Okay. Valacyclovir is a variant prodrug of the acyclovir and is particularly useful in treatment of VZV infection. When this product is given orally, blood levels of acyclovir is obtained, which are equivalent to those obtained by intravenous administration. Decyclovir is another product of acyclovir, but some more toxic and limiting in its potential. Ganciclovir is phosphorylated by thymidine kinase, product produced by both alpha beta subfamily of the subfamily of the herpes virus. So it can be used in both virus, but unfortunately, the drug is not safe as acyclovir because it can be incorporated into the cellular DNA. Nevertheless, it can be used for the treatment of the CMV or cytomegalovirus, okay, infection. This virus, which is also causes eye infection and lead to the blindness. Acyclovir is not effective to the infection because CMV does not encode the viral thymidine kinase. This is important. Okay, acyclovir cannot, cannot be used for CMV because CMV does not encode a viral thymidine kinase. Acyclovir, however, can be converted to monophosphate by kinases other than the thymidine kinase. Yeah, this is lower bioavailability and the variant port drug can be introduced to treatment of CMV infection. Panciclovir has essentially the same spectrum of activity and but has a better potency, faster onset, and longer duration of action. It is used topically for the treatment of the cold sores and intravenously for the treatment of HSV in the immunocompromised patients. Like acyclovir, penciclovir also has a poor bioavailability and poor absorb from the gut because of this polarity. Okay. Then we have the cidoclovir, which is a broad-spectrum antiviral agent which shows selectivity for viral DNA polymerase and used to treat the retinal infection caused by the CMV. So, okay, so this is, but unfortunately, the drug is extremely polar and has a poor oral bioavailability and toxic for kidneys. Edoxudine can be used for the topical treatment of the herpes schistosomiasis, and trifluridine is a drug of choice for the disease because it is effective for the lower dose. And the vidarabine is the early antiviral drug, but acyclovir is now generally preferred. Foscarnet is used for the treatment of CMV retinitis when it is approximately equal to activity of the ganciclovir. It can also be used in the immunocompromised patient for the treatment of HSV and VZS strain proof to the acyclovir, but it does not undergo the metabolic activation. Okay, so among all these drugs, foscarnet is the drug that can be used for the CMV retinitis. Cyclovir also can be used, but acyclovir cannot be used for the drug because acyclovir, we all know first it needs to get activated by this viral thymidine kinase where the first phosphorylation happens. Okay, but it is not possible in CMV. That is why acyclovir cannot be used for this CMV retinitis. Now I will go to the next problem. This is the structure of foscarnet. Okay. So problem number 7 says which enzyme is responsible for the SCID or severe combined immunodeficiency syndrome? Your options are number A catechol dehydrogenase, number B cytochrome C oxidase, number C adenosine deaminase, and number D adenylate deaminase. Which of the following enzyme is responsible for SCID or severe combined immunodeficiency syndrome? Which enzyme is responsible for SCID or severe combined immunodeficiency syndrome? Catechol dehydrogenase, cytochrome C oxidase, adenosine deaminase or adenylate deaminase? Let me know A, B, C, or D. Which of the following will be the correct answer for problem number 7? I will check your check your answers after a minute. Okay. Which enzyme is responsible for the SCID? Let me check your answers first. Okay. I have received two answers and both are saying that number C is the correct one. Okay, let me check if number C is the correct answer or not. So the correct answer is very good. Number C, adenosine deaminase is the enzyme which is responsible for the SCID. So basically, what does this adenosine deaminase do? It converts the adenosine to the inosine. Okay. So here you can see this amine NH2 is present in case of the adenosine, but it is converted to this ketone. So this is what does the deaminase does. It removes the NH2. So severe combined immunodeficiency or SCID consists of a group of genetic disorder characterized by a block in T lymphocyte differentiation that is variably associated with the abnormal development or other lymphocyte lineages. What is the B or NK lymphocyte or more rarely of the myeloid lineage. A genetic disorder in which both arms of the B and T cells of the adaptive immune system are impaired due to the defect in the several possible genes. SCID is a severe form of the heritable immunodeficiency. In overall frequency, it is estimated 1 in 75,000 or 1 lakh births. Okay. The pig molecule surrounded by the protein can be viewed as a kind of the hydrophilic and polymeric shield for both protein and disguise the protein. The PEG polymer has the advantage that it shows a little toxicity. The enzymes L-asparginase and adenosine deaminase have been treated in this way to grieve the protein PEG conjugate called the pegasparase and pegdiamase, which has been used for the treatment of the leukemia and SCID, the severe combined immunodeficiency syndrome, respectively. SCID is the immunological defect associated with the lack of the adenosine deaminase. The conjugates have a longer plasma half-life and the enzyme alone are the less likely to produce immune response. Interferon has similarly been pegylated to form a preparation called the peginterferon alpha-2 beta to alpha-2 b, that which is used for the treatment of the hepatitis C. So it is the one example how the pegylation happens in case of this kind of disease. Okay. So remember SCID, which is a very severe immune deficiency disease, is causes because of the enzyme this adenosine deaminase. Okay. Now let me go to the next problem. Problem number 8 says statins works as, your options are number A, HMG-CoA synthase inhibitor, number B, HMG-CoA reductase inhibitor, number C, squalene synthase inhibitor, and number D, acetoacetyl-CoA thiolase inhibitor. Statins work as which of the following? HMG-CoA synthase inhibitor, HMG-CoA reductase inhibitor, squalene synthase inhibitor, or acetoacetyl-CoA thiolase inhibitor. You have to fill in the blanks. A, B, C, or D. Let me know which of the following will be the correct answer. Statins work as which of the following? HMG-CoA synthase inhibitor, HMG-CoA reductase inhibitor, squalene synthase inhibitor, or acetoacetyl-CoA thiolase inhibitor. I will wait. I will check your answers after a minute. Okay. Let me know. A, B, C, or D. Which of the following will be the correct answer? Statins work as which of the following? HMG CoA synthase inhibitor, HMG CoA reductase inhibitor, squalene synthase inhibitor, or acetoacyl CoA thiolase inhibitor? Statins work as which of the following? Let me check your answers first, then I will discuss it. Okay. Okay, number B, number B. So I have received two answers and both are saying that number B is the correct one. Okay, let me find out if number B is the correct answer or not. The correct answer is yes, number B, HMG CoA reductase inhibitor. So, HMG-CoA reductase inhibitors are also known as statins, are a class of the lipid-lowering drugs that reduce the illness and mortality in those who are at a high risk of the cardiovascular disease. They are the most common cholesterol-lowering drugs. Okay, so this HMG-CoA inhibitors or this kind of the statins are the cholesterol-lowering drugs. Okay, they reduce the risk of the illness related to the arteriosclerosis, where the varying degree of the effect depending on the underlying risk factor and history of the cardiovascular disease. Clinical practice guidelines generally recommend people start with a lifestyle modification through the cholesterol-lowering diet and physical exercise. Mechanism: HMG-CoA reductase pathway, which is blocked by the statins by inhibiting the rate-limiting enzyme HMG-CoA reductase. Okay, statins act by competitively binding to the HMG-CoA reductase and the rate-limiting enzyme of the mevalonate pathway. Because statins are similar to the structure of HMG-CoA on molecular level, they will fit into the enzyme's active site and compete with the native substrate, which is the HMG-CoA. This competition reduces the rate by which HMG-CoA reductase is able to produce the mevalonate, the next molecule in the cascade, and eventually produces the cholesterol. So this in this way, it actually hinders the cholesterol biosynthesis in the body. So these are the examples of the statins which are, which are used as a cholesterol lowering drug. The green one, green means this is the year when the drug is approved from the FDA, and the red is the pharmacophoric group, okay, which is the important for the activity of the drug. So this is the pravastatin. Again, I am saying the you do not need to remember the structures, the names that you need to remember, okay. Pravastatin, then we have simvastatin, we have the fluvastatins, atorvastatins, rosuvastatin, and pitavastatins, and then we have the mevastatin and lovastatin. All these have a suffix of the statin that will help you to remember that these are the cholesterol lowering drugs. Okay, so so these are the example of the HMG CoA reductase pathway. Or there are more enzymes actually in this biosynthetic pathway of the cholesterol biosynthesis, and each and every enzymes can be inhibited to stop this particular biosynthetic pathway. But statins and HMG CoA, the HMG CoA reductase inhibitors are the most important ones in this particular therapeutic area of the cholesterol lowering drugs. So let me go to the problem number 9. It says that oseltamivir is a dash agent. Your options are number A, anti-influenza, number B, anti-herpes virus, number C, antiretroviral, and number D, antifungal. Oseltamivir is a dash agent. Your options are anti-influenza, anti-herpes virus, antiretroviral, or antifungal. Let me know A, B, C, or D, which of the following will be the correct answer. Oseltamivir is which kind of agent? I want all of you to answer. Okay, even if your answer is wrong, it's fine, but try to answer the questions. Oseltamivir is which kind of agent? Anti-influenza, anti-herpes virus, antiretroviral, or antifungal? I will check the chat box after 2 minutes. Meanwhile, let me know A, B, C, or D, which of the following you think is the correct answer for problem number 9. Oseltamivir is which kind of agent? Let me check your answers. Okay. Number C, number A, and number A. So I think I have received two number A and one number C. Hmm. Okay, let me check which of the following will be the correct answer. The correct answer is number A, anti-influenza. Oseltamivir is a anti-influenza agent. Okay, so this is the structure of the oseltamivir. So this is also known as the brand name of the Tamiflu. Okay, it is an antiviral medication used to treat or prevent influenza A and B. It belongs to a class of the drugs called the neuraminidase inhibitor. This is important. This is something new. Okay, so this is the oseltamivir is an example of the now it falls in the class of the neuraminidase inhibitor, which work by trapping newly formed virus particle inside the infected cell. So okay, and preventing them to spreading throughout the body. Mechanism of action, prodrug activation. Oseltamivir is an ester product. After oral intake, it is rapidly converted in the liver by the enzyme human carboxyl esterase or HCE1 into its active form, oseltamivir carboxylate. Viral blocking, the active carboxylate metabolite competitively inhibits the viral enzyme neuraminidase. This enzyme is responsible for cleaving the sialic acid on the host cell surface. By blocking it, the drug prevents the release of the progeny variants. Okay. Resistant mechanism. The most significant challenge for oseltamivir is the emergence of the resistant strain. The H275Y mutation, the most common resistant mutation in the N1 subtype like swine flu or H1N1 is the H275Y histidine to ribosome substitution. This mutation prevents the enzyme from undergoing the necessary conformational change to accommodate the oseltamavir's bulky side chain, reducing the binding affinity by roughly 300-fold. E119V mutation found in some H3N2 and H7N9 strain. This mutation decreases the conformational stability and drug enzyme complex, leading to lower efficacy and fitness cost, while H27 5Y mutation confers resistant. It often carries a fitness cost map, meaning the resistant virus may replicate slower and spread less efficiently than the wild type of the virus in absence of the drug. Okay, so this is the importance and all about the oseltamivir. Now I will go to the problem number 10. It says that pivamcillin is a prodrug of dash. You have to fill in the blanks. Your options are number A, penicillin, number B, cephalosporin, number C, ampicillin, and number D, amoxicillin. Pivamcillin is a prodrug of which of the following? Penicillin, cephalosporin, ampicillin, or amoxicillin? Let me know A, B, C, or D. Which of the following will be the correct answer? Pivamcillin is a prodrug of which of the following? Penicillin, cephalosporin, ampicillin, or amoxicillin? Pivampicillin is a prodrug of which of the following? Penicillin, cephalosporin, ampicillin, or amoxicillin? Let me check the chat box. Okay, number C, number C. So I have received number C as the answer for this question number 10. Okay, let me check. So yeah, number C is the correct answer. Ampicillin is the Pivampicillin is a prodrug of ampicillin. So ampicillin prodrugs. So pivampicillin, talampicillin, and vacampicillin are the prodrugs of the ampicillin. In all these three cases, esters used to mark the carboxylic acid group seem rather elaborate, and one may ask why the simple methyl ester cannot be used. The answer is that methyl esters of penicillins are not metabolized in humans. The bulky penicillin skeleton is close to the ester, that is, acts as a steric shield and prevents the esterase enzyme to catalyze the reaction to form accepting the penicillin ester for the substrate. Fortunately, acyl methoxy esters are susceptible to the esterases. These extended esters contain the second ester group further away from the penicillin nucleus and which is more exposed to attack. The hydrolysis products are inherently unstable and decompose spontaneously to release the formaldehyde and reveal the free carboxylic acid. The release of the formaldehyde is not ideal and is a toxic chemical. However, it is formed naturally in the body through the enzymatic demethylation of the various compounds found in the diet, and the levels produced from the prodrugs described cause a little problem. Moreover, the drugs are only taken in the short period of time. Such extended ester can be used to prepare prodrugs of the penicillin, but one has to be careful as one does not go to further extreme and make the penicillin too lipophilic. For example, one acyloxy ester of penicillin G is so lipophilic that it has a poor solubility in water. Fortunately, this problem can be avoided by making the extended ester more polar, like for attaching the valine or something. So these are the prodrugs are used to aid the absorption of penicillin to the gut wall. Pivampicillin when R equal to when this R equal to this group, it is the pivampicillin. When this is not talampicillin, and this is the vacampicillin. Okay, so the mechanism by which the acyl methoxy esters are hydrolyzed is this one. So this is the penicillin, and we have this kind of group. So this is the esterase if it comes here. So by which we have another ester group is here, and formaldehyde will be released, and mechanism, and this is the polar extension of the valine group, which is the polar extended for ester of the penicillin G. Okay. So pivampicillin is a prodrug of the ampicillin. Now I will go to the next problem. Problem number 11 says, which of the following is not an example of antimetabolites used in antibacterial therapeutics? Your options are number A, trimethoprim, number B, doxycycline, number C, sulfone, and number D, sulfamethoxazole. Which of the following is not an example of antimetabolites used in the antibacterial therapeutics? Trimethoprim, doxycycline, sulfone, or sulfamethoxazole? Or sulfamethoxazole. Which of the following is not an example of antimetabolites used in the antibacterial therapeutics? Let me know A, B, C, or D. Which of the following will be the correct answer? Which of the following is not an example of antimetabolites used in the antibacterial therapeutics? Which of the following is not an example of antimetabolites used as the antibacterial therapeutics? Trimethoprim, doxycycline, sulfone, or sulfamethoxazole? I will check the chat box after a minute. Let me know A, B, C, or D. Which of the following will be the correct answer of problem number 11? Which of the following is not an example of antimetabolites used in the as the antibacterial therapeutics? Let me check your answers. Okay, number C, number C, number C. So I have received all the answers as number C. Let me check if number C is the correct answer or not. So the correct answer will be it is number B. Doxycycline is not an example of the antimetabolites used as the antibacterial therapeutics. Okay. So now let us discuss about the antimetabolites. Okay, so trimethoprim is the orally bioactive diamino-pyrimidine structure. So this is the structure of the trimethoprim. It has a pyrimidine group which has two amine groups. So it is a diamino-pyrimidine structure. Okay, which is proved to be highly selective antibacterial and antimalarial agent. It acts against the dihydrofolate reductase. Yesterday, when I was discussing about this, your folic acid metabolism in the antibacterial. I have mentioned that these enzymes are very important in the pathway, and trimethoprim is the enzyme. Trimethoprim is the drug, actually, the agent which inhibits this enzyme. Okay, so it inhibits the dihydrofolate reductase, an enzyme which carried out the conversion of dihydrofolate to tetrahydrofolate, leading to the inhibition of the DNA synthesis and cell growth. Okay. The dihydrofolate reductase is present in the mammalian cells as well as the bacterial cell, but mutation over millions of years have resulted significant difference of the structure between these two enzymes such that the trimethoprim recognizes and inhibits only the bacterial enzyme strongly. In fact, trimethoprim is 1 lakh times more active against the bacterial enzyme. Trimethoprim is often given in conjugation with the sulfonamide like sulfamethoxazole, and the preparation is known as the clotrimazole that I have discussed yesterday itself, and it is an example of your sequential blocking, right? We have discussed the sequential blocking when two different drugs they actually used to inhibit a same biosynthetic pathway then two different point. Clotrimazole is the drug preparation which have this trimethoprim and along with this sulfonamide sulfomethoxazole, and this inhibits the incorporation of PABA to dihydrofolate, and while trimethoprim inhibits the dihydrofolate reductase. Therefore, two enzymes in one biosynthetic route is inhibited. This is a very effective method of inhibiting the biosynthetic route and has an advantage for the doses of the various drug that too can be kept down the safe level. To get same level of inhibition using a single drug, the dose level would have been much higher, leading to the possible side effect. This is known as I was saying, this is a sequential blocking. Okay. Resistance to trimethoprim has been observed in E. coli and which produce a new form of the target enzyme that has a less affinity to the drug. The sulfones are the most important drugs used in the treatment of leprosy. It is believed that they inhibit the same bacterial enzyme inhibited by the sulfonamides. Okay, so these drugs, these trimethoprim and sulfones and sulfametrazole, all are the examples of the antimetabolites that are used as an antibacterial agent, but not the doxycycline. Okay, doxycycline is not an example of the antimetabolites because we know doxycycline is falls in a different class of the antibacterial agents. Okay. Now I will go to the next problem. Problem number 12 says which of the following reactions is used in the dynamic combinatorial synthesis of the vancomycin dimers. Your options are number A Suzuki coupling, number B Bergman cyclization, number C olefin metathesis, and number D Michael reaction. Which of the following reactions is used in the dynamic combinatorial synthesis of the vancomycin dimer? Suzuki coupling, Bergman cyclization, olefin metathesis, or Michael reaction? Which of the following reactions is used in the dynamic combinatorial synthesis of vancomycin dimer? Suzuki coupling, Bergman cyclization, olefin metathesis, or Michael reactions? Let me know A, B, C, or D. Which of the following will be the correct answer of problem number 12? I will check your answers after a minute or two. Okay, let me know. Which of the following reactions is used in the dynamic combinatorial synthesis of vancomycin dimers? Let me check the chat box. Let's see if there is any answer for the problem number 12. Okay, number B. So I have received two answers and both are saying that the number B is the correct one. Okay, let me check if number B is the correct answer or is there any different answer which is correct. The correct answer is it is number C. Okay, olefin metathesis is the reaction which is used in the synthesis, the combinatorial synthesis of the vancomycin dimers. So, what is the dynamic combinatorial synthesis? So, vancomycin is basically an antibiotic that works because it marks the building block required for the bacterial cell wall synthesis. Binding takes place specifically between the antibiotic and the peptide sequence L-lysine DLA, which is present in the building block. It is also known that this binding promotes dimerization of the vancomycin target complex, which suggests that covalently linked vancomycin dimer might be more effective antibacterial agents than vancomycin itself. A dynamic combinatorial synthesis was carried out to synthesize a variety of the different vancomycin dimer covalently linked by bridges of the different lengths. The vancomycin monomers used had been modified such that they contain long chain of alkyl substituents with double bonds at the end. The reaction between two double bonds in present catalyst lead to the bridge formation is known as the olefin metathesis. So this is the olefin metathesis. We have two double bonds here, and then the formation on this kind of catalysts are used in case of the olefin metathesis. Okay, the tripeptide target was present. So here you can see these are the vancomycin monomers, and this is the tripeptide. So the tripeptide target was present to accelerate the rate of the bridge formation and to promote the formation of the vancomycin dimer having ideal bridge length. The vancomycin monomers bind the tripeptide, which encourages the self-assembly of the non-covalently linked dimer. Once formed, these dimers having correct lengths of the substituent are more likely to react together to form the covalent bridge. Having established the optimum length of the bridge, another experiment was carried out on the 8 vancomycin monomers, which have the correct length of the tether but varied slightly to the structure. The mixture of 36 possible products was analyzed by the mass spectrometry to indicate the relative proportion of each dimer formed. 11 of 36 compounds were then synthesized separately, and it is found that the antibacterial activity matched with the level of amplification, that is, the compound present is greater quantity than the activity. So this is the tripeptide, and this is the structure of the vancomycin monomer. The first is the ligand binding, then the dimerization happened, then at the end, as you can see, this is the example of the olefin metathesis, which give rise to the covalently linked vancomycin dimer. Okay, so the reaction which is involved in the dynamic combinatorial synthesis of this vancomycin dimers is the olefin metathesis. Now I will go to the next problem. Problem number 13 says, according to fragment-based drug discovery, which of the following parameters are incorrect for the fragments to be used in the lead compounds? Your options are number A molecular weight greater than 300, number B, no more than 3 rotable bonds, number C, LC log P value equal to 3, and number D, no more than 3 hydrogen bond donor and acceptor. According to fragment-based drug discovery, which of the following parameters are incorrect for the fragments to be used in the lead compounds? Your options are number A molecular weight greater than 300, number B, no more than 3 rotable bonds, number C, C log P value equal to 3, and number D, no more than 3 hydrogen bond donor or acceptor. Which of the following parameters are incorrect for the fragments to be used as in the lead compound? Let me know A, B, C or D, which of the following is the correct answer for problem number 13. So, which of the following parameters are incorrect for the fragments to be used in the lead compounds according to fragment-based drug design? Let me check your answers first. Okay, number B, number C. So I have received three answers, number D, number C, and number D. So three different options as the answer. Okay, let's find out which of the following will be the correct answer, and let us discuss about the fragment-based drug discovery. The correct answer is number A molecular weight greater than 300. Okay, this statement is incorrect. Now let us understand. Properties of a lead compound. Some of the lead compounds have been isolated from the natural sources and have insufficient activity to be used. Okay, 1 second. Yeah. To be used directly to the medicine without the serious side effect. And for example, morphine, quinine, paclitaxel. However, most lead compounds have low activity and or unacceptable side effects, which means significant amount of structural modifications are required. If the aim of the research is to develop an orally active compound, then certain properties of the lead compound should be taken into account. Most orally active drugs obey the rules laid by the Lipinski's rule of 5 or Weber's parameter. A study which is known as the orally active drugs and lead compounds from which they were derived demonstrated that the equivalent rules for the lead compound should be more stringent. This is because the structure of the lead compound almost certainly has to be modified and increased, both in terms of the size and hydrophobicity. The suggested properties of a lead compound are that it should not be molecular weight of 100 to 350 AMU, a C log P value of 1 to 3. C log P is a measure of how hydrophobic a compound is, and in general, there is an average increase of the molecular weight in AMU and increase of 1 C log P when going to the lead compound to final drug. Studies also show that the lead compound generally has a fewer aromatic rings and hydrogen bond acceptor compared to the final drug. Such consideration has to be taken into account when deciding the lead compound to use for the research project if several structures are available. Another approach is to making this decision is to calculate the binding and ligand efficiency of the potential lead compound. This has to be done dividing the free energy of the binding of each molecule by the number of the non-hydrogen bond atom present in the structure. The better ligand efficiency and the lower molecular weight of the final optimized structure is likely to be. Moreover, if you have a choice of the lead compound, most suitable ones necessary is not it is not necessary that the most suitable ones will be the most potent. For the fragment-based drug discovery, a rule of 3 has been suggested. So, rule of 5, we know for the Lipinski's rule of 5 for the oral bioavailability of the drug. But in case of the fragment-based drug discovery, there is a rule of 3. Now, so, what is this rule? So, a molecular weight less than 300, not more than 3 hydrogen bond donor, not more than 3 hydrogen bond acceptor, C log P equal to 3, no more than 3 rotable bonds, a polar surface area of 60 Armstrong square. So these are the rules that has to be followed in case of the fragment-based drug discovery. These are the parameters that has to be followed for a particular compound to be a good lead compound. Okay. So the statement number A, which says that the molecular weight greater than 300, this is incorrect. Okay. Now I will go to the next problem. Problem number 14 says, which of the following is not a semi-synthetic antibacterial agent? Now, your options are number A, ampicillin, number B, carbonicillin, number C, cephalexin, and number D, sulfonamide. Which of the following is not a semi-synthetic antibacterial agent? Ampicillin, carbonicillin, cephalexin, or sulfonamide? Let me know A, B, C, or D. Which of the following is the correct answer? Which of the following is not a semi-synthetic antibacterial agent? Ampicillin, carbonicillin, cephalexin, and sulfonamide. After a minute, I will check the chat box. Meanwhile, let me know A, B, C, or D. Which of the following will be the correct answer? Which of the following is not a semi-synthetic antibacterial agent? Number D, number C, number D, number A. So I have received, I think, okay, no, I have received number D, number C, and number D. So two number D and one number C I have received as answer. So C and D are the options. Okay, let me check. So the correct answer is number D. Yes, sulfonamides is not an example of semi-synthetic antibacterial agent. So the sources of, according to the sources, the antibacterial agents can be natural, mainly fungal sources. The original antibiotics were derived from the fungal sources. These can be referred as the natural antibiotics. For example, benzyl penicillin and gentamicin are the natural antibiotics. Semi-synthetic, chemically altered natural compound developed to decrease the toxicity and increase the effectiveness. Ampicillin, amikacin is a semi-synthetic antibiotic, synthetic, chemically designed in a lab. Chemical bacteria are not exposed to the compounds until they are released. They are also designed to have some greater efficiency and less toxicity, moxifloxacin and norfloxacin. Okay, so sulfonamides it is a synthetic antibacterial agent, not semi-synthetic, not natural. It is a synthetic, completely synthetic antibacterial agent that is designed, chemically designed and developed in the laboratory. So sulfonamides or sulfa drugs are the antibiotics that have been structural similarity with the para-aminobenzoic acid or PABA containing the sulfonamide groups in their chemical structure. They have bacteriostatic action against the gram-positive and gram-negative bacteria. For example, sulfadazine, sulfamethoxazole, cotrimoxazole, or trimethoprim. Mechanism of action: they compete with the para-aminobenzoic acid. Is an essential precursor of the synthesis of folic acid required for the synthesis of DNA and RNA in bacteria. They inhibit the bacterial folate synthesis, leading to the impairment of the DNA formation. Okay. So this is the structure of the para-aminobenzoic acid. So see how it binds to the active site. The phenyl group leads to a Van der Waals interaction where NH2 and COOH. NH2 forms a hydrogen bonding and COO minus group form a ionic bond. Sulfonamide similarly they form the Van der Waals interaction, hydrogen bonding and ionic bonding. That is why they are an example of the competitive inhibitor. What is a competitive inhibitor? We all know that it has a structural similarity with our natural that normal host of the particular enzyme, and because of that it competes with the natural substrate of a particular enzyme to the active side. Okay. So it is an example. So sulfa drugs, which are the completely synthetic antibacterial agents, are very important, and they are a competitive inhibitor in nature. Okay. Now, problem number 15 says, in the TCA cycle, the conversion of malate to oxaloacetate is catalyzed by which of the following enzymes? Your options are number A, malate dehydrogenase, number B, malate dehalogenase, number C, oxaloacetate synthase, and number D, aconitase. In a TCA cycle, the conversion of malate to oxaloacetate is catalyzed by which of the following enzymes? Malate dehydrogenase, malate dehalogenase, oxaloacetate synthase, or aconitase. Let me know A, B, C, or D, which of the following will be the correct answer of problem number 15. I will check the chat box after a minute. In TCA cycle, conversion of malate to oxaloacetate is catalyzed by which of the following enzyme? Malate dehydrogenase, malate dehalogenase, oxaloacetate synthase, or aconitase. In the TCA cycle, the conversion of malate to oxaloacetate is catalyzed by which of the following? What will be the correct answer? Okay, number A, number A, number A. So I have received one number C and others are saying that number A is the correct answer. Okay, let us find out. So the correct answer will be number A. Malate dehydrogenase is the enzyme which converts the malate to oxaloacetate in a TCA cycle. So the malate to oxaloacetate, as you can see, the conversion is the malate dehydrogenase. So the dehydrogenation reaction happens here. Okay, two hydrogens are eliminated and NAD plus is the co-substrate is there which is converted to NADH and H plus. Okay, so this is the TCA cycle, the fumarase enzyme activity. So it starts with the pyruvate which is converted to acetyl CoA, and then from citrate we have this isocitrate with the help of enzyme aconitase. Isocitrate is converted to oxalosuccinate with the isocitrate dehydrogenase. Oxalosuccinate is converted to alpha glutoglutarate with the isocitrate dehydrogenase, which is converted to succinyl CoA with the alpha glutoglutarate dehydrogenase. Then succinyl CoA goes to succinate with the succinyl CoA synthase. Then succinate converted to fumarate with the succinate dehydrogenase. Fumarate goes to malate with the fumarase, and malate goes to oxaloacetate with the malate dehydrogenase. And all these complete cycles, we have a couple of NaH, NAD plus, FAD, and all this as the co-substrate or coenzymes. Okay. So yeah, malate dehydrogenase is the correct answer. Now problem number 16 says which of the following statements is true regarding the HIV protease enzyme? Your options are number A, it belongs to enzyme family of the cysteine protease. Number B, it catalyzes the formation of the peptide bonds. Number C, active site structure contains cysteine moiety. Number D, active site structure contains the aspartic acid. Which of the following statements is true regarding the HIV protease? Number A, it belongs to an enzyme family of the cysteine protease. Number B, it catalyzes the formation of the peptide bonds. Number C, the active site structure contains the cysteine moiety. Number D, active site structure contains the aspartic acid. Which of the following statements is true regarding the HIV protease enzyme? Read the options carefully and then answer. Let me know in the chat box A, B, C or D which of the following is the correct answer for problem number 16. Which of the following statements is true regarding the HIV protease enzyme? Let me check the chat box. Okay, number A, number D. I think I have, okay, number A. Hmm. Okay, so this is number 6 for number 16, right? So number D and number A, two are the answers that I have received till now. Okay, let me check if both of our, which of the following is the correct answer. So the statement which is true will be number D, which says that the active site structure contains the aspartic acid. This statement is true, okay? So HIV protease enzyme is an example of an enzyme family called aspartyl protease, enzymes which catalyze the cleavage of the peptide bonds and which contain aspartic acid in the active site and is crucial for the catalytic metabolism. The enzyme is relatively small, can be obtained by synthesis. Alternatively, it can be cloned and expressed in the fast-growing cells, then purified in large quantities. The enzyme is then crystallized with the or without inhibitor bound to the active site, making it ideal target for the structure-based drug design where the where the crystallographic study of the enzyme inhibitor complexes are allowed to design the novel inhibitor. So the HIV protease is a symmetrical dimer made up of two identical protein subunits, each consisting of 99 amino acids. The active site is an interface between the protein subunit is also symmetrical with the two-fold rotational C2 axis. The amino acids aspartate 25, thiothionine 26, and glycine 27 from each monomer are located on the floor of the active site, and each monomer provides a flap to act as a sealing. The enzyme has a broad substrate specificity and can be cleave a variety of peptide bonds in viral polypeptide, but critically it can cleave both bonds between a proline residues and with an aromatic residue, like phenylalanine or tyrosine. The cleavage of the peptide bond next to the proline are unusual but does not occur in the mammalian protease, such as renine, pepsin, but so chances of achieving selectivity against HIV protease over mammalian protease are good. Moreover, the symmetrical nature of the viral enzyme and active site are not present in the mammalian protease, suggesting the possibility of drug selectivity. That is why HIV protease enzyme is a very good target for the developing the anti-HIV drugs. These are the examples. You don't need to remember the structures, but remember the names. Okay, indinavir, nelfinavir, ritonavir, saquonavir, and ampranavir. All these are examples of the HIV protease enzyme inhibitors. Okay. Now, problem number 17, it says which of the following statements regarding RNA is false? Your options are number A, rRNA acts as a catalyst to make the peptide bond. Number B, the amino acid sequence of proteins depend on the base sequence of mRNA. Number C, RNA polymerase 3 synthesizes the mRNA. Number D, RNA polymerase 1 synthesizes the rRNA. Which of the following statement is false? Number A, rRNA acts as a catalyst to make the peptide bond. Number B, the amino acid sequence of proteins depend on the base sequence of mRNA. Number C, RNA polymerase 3 synthesizes mRNA. And number D, RNA polymerase 1 synthesizes rRNA. Which of the following statement is false? Let me know A, B, C or D. Which of the following will be the correct answer for problem number 17? Let me check the chat box. Let's see what is the answers that are given. Number C. So I have received two answers and both are saying that number C is the correct answer. Okay, fine. The correct answer will be yes, number C, RNA polymerase 3 synthesize mRNA. This statement is false. So there are three types of RNA polymerase 1, 2, and 3. RNA polymerase 1 is that only transcripts ribosomal RNA or rRNA, but not 5S rRNA, okay. And RNA polymerase 2, central enzyme that catalyzes DNA-directed mRNA synthesis during the transcription of the protein coding genes. And RNA polymerase 3 is the that transcribes small translated RNAs such as the tRNA. Transcribes RNA genes is the RNA polymerase 1. RNA polymerase 2 transcribes the mRNA, miRNA, and snRNA and snRNA genes. RNA polymerase 3 transcribe for the tRNA and 5S rRNA genes. RNA polymerase 1 resides in the nucleons. The RNA polymerase 2 resides in nucleoplasm, and RNA polymerase 3 also resides in nucleoplasm. RNA polymerase 1 transcribes pre-rRNA for the 2200 copy. The RNA polymerase 2 transcript to around 25,000 genes, and RNA polymerase 3 transcribe 30 to 50 genes at a variable copy numbers. RNA polymerase 1 contain the same two non-identical alpha-like subunit. RNA polymerase 2 has two copies of different alpha-like subunit, and RNA polymerase 3 contain the same two non-identical alpha-like subunit. RNA polymerase 1 are insensitive to alpha amantinin, amanitin. RNA polymerase 2 sensitive to 1 microgram per ml alpha amanitin, and RNA polymerase 3 sensitive to 10 microgram per ml alpha amanitin. Okay, so this is the differences between the RNA polymerase 1, 2, and 3. Problem number 18 says which of the following lead compound is not derived from the microorganism? Your options are number A cyclosporine, number B curacacin A, number B lipstyrene, and number D rasfonin. Now, which of the following lead compound is not derived from microorganism? Cyclosporine, curacacin A, lipstyrene, or rasfonin? Let me know. A, B, C, or D. Which of the following is the correct answer? Which of the following lead compound is not derived from microorganisms? Cyclosporine, curacacin A, lipstyrene, or rasfonin? Which of the following lead compound is not derived from microorganism? Let me check the chat box. Let's see if there is any answer for problem number 18. Okay, number B, number C, number C. So I have received two number C's as the answer and one number B. Let me check which of the following is the correct answer. So the correct answer will be number B. Curacacin A is not derived from microorganisms. Okay. So microorganisms. Okay, let's first understand. So this cyclosporine, lipstyrene, and rasforin, or these lead compounds are derived from the microbiological sources or microorganisms, such as the bacteria and fungi, which have been provided a rich packing pickings of the drugs and lead compounds. These organisms produce a large variety of antimicrobial agents which have evolved to give their host an advantage over their competitors in the microbiological world. The screening of the microorganism became highly popular after discovery of penicillin. Soil and water samples were collected from all around the world in order to study new fungal and bacterial strain, leading to impressive arsenal of the antibacterial agents such as the cephalosporin, tetracycline, aminoglycoside, rifamycin, and chloramphenicol and vancomycin. Although most of the drugs derived from the microorganism are used in the antibacterial therapy, some microbial and metabolites have provided the lead compound in other fields of medicine. For example, aspergillosin is isolated from the Aspergillus allius is a novel antagonist for the peptide hormone called the CCK or cholecytokinin. This is involved in the control of the appetite. CCK is act as a neurotransmitter in the brain and through involved in the panic attacks, although the aspergillus may or therefore have potential to targeting anxiety. Other examples are the lovastatin, which is the first clinically used statins to lower the cholesterol, and another fungal metabolite is the cyclosporin, which is the suppress the immune response after organ transplant. Lipstatin is the natural product which has isolated from from the source. It inhibits the pancreatic lipase and was when it inhibits the pancreatic lipase and was the lead compound for the anti-obesity compound or listed. And finally, a fungal metabolite called rasponin, which promotes the cell death in the cancer cells, not normal cells. This represents a promising lead compound for the novel anti-cancer agent. Now, the age drug, which is the curacacin A, it is not derived from the microorganism, but it is derived from the marine source. In recent years, a great interest in finding lead compounds from the marine sources, coral, sponges, fish, and marine microorganism have a wealth of biologically potent chemicals with an interesting inflammatory, antiviral, and anti-cancer activity. For example, curacacin A is obtained from the marine cyanobacterium and shows the potent anti-tumor activity. Other anti-tumor agents derived from the marine sources include the this all these examples. Okay, and so this is the structure of the curacacin A. Okay. Now, problem number 19, it says which of the following heterocyclic rings is present in cimatidine? Your options are number A, imidazole, number B, pyrazole, number C, indole, and number D, benzimidazole. Which of the following heterocyclic rings is present in cimatidine? Imidazole, pyrazole, indole, or benzimidazole? Which of the following heterocyclic rings is present in cimatidine? Let me check the chat box. Okay, number A. So I have received two answers and both are saying that number A is the correct answer. Okay, let me find out which of the following is the correct answer. Okay, very good. Imidazole is the ring which is present in the cimatidine drug. Okay, so these are the examples of the aromatic heterocyclic compounds. Okay, so the names, try to remember all the names of the heterocyclic compounds, at least this few of these. So this is the structure of the cimatidine. It has the imidazole ring, and this is the synthetic procedure. Okay, so synthesis of cimatidine was originally carried out as a four-step process where the lithium aluminium hydrate was used as a reagent for the initial reduction step. Subsequent research revealed that the reduction could be carried out more cheaply and safely using sodium in liquid ammonia. So this become method used in the manufacturing process. So this is the lithium for after the this is the ester upon the usage of the lithium aluminium hydride. The or this condition reduction happens to create an alcohol, and then this kind of thiol comes, and this is the substitution reaction, and then for further reaction gives us the cimetidine. The cimetidine inhibits the gastric acid release by acting as an antagonist of the H2 receptor. It does not show the toxic side effect observed in the metamide, and has shown to slightly more active. It has also found it to inhibit the pentagastrin from stimulating release of the gastric acid. Pentagastrin is an analog of the gastrin, and in fact the cimetidine inhibits it suggests some relationship between the histamine and gastrin in the release of the gastric acid. Cimetidine was first marketed in UK in 1976 under the name of the Tegamet, and it was the first orally active anti-ulcer drug doing away the need of the surgery. For several years, it was world's highest selling production and until it pushed to the second place by ranitidine. Okay. Now, last question, it says which of the following statements is false regarding NRTIs? Your options are number A, they have a good oral bioavailability. Number B, they act on both HIV 1 and 2. Number C, they are less toxic than NNRTIs. And number D, they are excreted through kidneys. Which of the following statement is false regarding NRTI? Number A, they have good oral bioavailability. Number B, they act against both HIV 1 and 2. Number C, they are less toxic than NRTIs. And number D, they are excreted through kidneys. Let me know which of the following is the statement is false regarding NRTIs. Which of the following statements is false regarding the NRTIs? Number A, they have a good oral bioavailability. Number B, they act against HIV 1 and 2. Number C, they are less toxic than NNRTIs. And number D, they are excreted through kidneys. Let me check the chat box. Okay. Number A, number B, number A. So I have received three answers, then two number A and one number B. So let us find out which of the following will be the correct answer. The correct answer is number C. They are less toxic than NNRTI. This statement is false. Okay. So basically, NRTI means nucleoside reverse transcriptase inhibitor. These are currently used as a part of the combination therapy in combating the HIV. Generally, NRTIs have a good oral bioavailability and are bound to minimally to the plasma protein and are excreted through the kidneys. They also act against both HIV 1 and 2. However, they are often associated with the toxic side effect. Zirobudin was the first HIV drug to reach the market and it can cause severe side effects such as the anemia. Lidonicine was the second anti-HIV drug approved for use and reached the US market in 1988. However, there is a risk of the toxicity of the pancreas. Abacavir was approved in 1998 and used successfully in children and combination with the protease inhibitor nelfanavir and saquanavir. However, life-threatening hypersensitivity reactions have been reported in some patients. Tenofovir disoproxil was approved for treatment of 2001. It remains in fact it's longer than many other antiretroviral drug, allowing once a daily dosing and can have toxic side effects on kidneys. It can be used alongside the emtricitabine, which is relatively free from the toxic side effect. Other NRTI used against HIV are the lamivudine and stavudine. Lamivudine is less toxic than zirobudine and has been approved for the treatment of the hepatitis B. Zalcotabine is the NRTI which against active against the hepatitis B, but long-term toxicity means it is unacceptable for the treatment of the chronic viral diseases which are not life-threatening. Adefovir was approved by US Food and Drug Administration of FDA in 2002 treatment of the chronic hepatitis B on the active virus such as the CMV and herpes. Non-nucleoside reverse transcriptase inhibitor on NNRTI compared to the NNRTI show a higher selectivity for HIV reverse transcriptase over the host DNA polymerase. As a result, these are less toxic and have fewer side effects. Unfortunately, the rapid resistance emerges if the NNRTI are used on its own, but this does not occur as it is combined with NRTI and the statement of treatment. NRTIs are restricted to HIV-1 actively and generally metabolized by the liver. They can interact with the drugs and bind more strongly to plasma protein, evaporin, efavirenz, and these are the examples of the NNRTI which are currently approved by the FDA for the treatment of the HIV. Okay, so this was the last question of today's session. Yesterday, somebody was asked has asked to how to remember different antibiotics because there are so many types of antibacterial drugs which have different mechanisms of action. So, so basically, there are several. So these five are the major classes. Okay, and there are different generations. You do not need to memorize the structure, but you have to understand the basics of the. I mean the pharmacophoric structure. For example, this is a penicillin. The penicillin has a four-member ring fused with a five-member nitrogen sulfur-containing ring with a beta-lactam. So this is the penicillin. Penicillin G, amoxicillin, ticarcillin, piperacillin, all are the examples of the penicillin. Okay. Then we have the cephalosporin, where this beta-lactam ring is combined with a six-member ring. So it is a common structure of the cephalosporin. Then we have the quinone, which has this kind of structure, and these are the example like nalidixic acid, ciprofloxacin, levofloxacin, and moxifloxacin. Macrolide, which has this structure, which has a very big cycle, so which is a macrolide, it's like erythromycin, clarithromycin, telithromycin, and tetracycline, which are four rings are fused together. That is why these are known as the tetracycline, and these are the examples of the classes. So this is this figure this is one of my very favorite figures for the mechanism of actions for the antibacterial drugs. So and it is very easy to remember. So there are several mechanisms which are all depicted in a single figure. First is a cell wall synthesis. Cell wall synthesis can be inhibited by these drugs. So we have cycloserine, vancomycin, bactricin, penicillin, cephalosporin, monobactam, carbamazepam. Okay. Then we have this is the cytoplasmic membrane and this is the cell wall and this is the we have this folic acid metabolism from PABA to DHFA to THFA. This I have discussed in today's class itself. The sulfonamides, they inhibit this conversion of PABA to DHFA, and we have this trimethoprim which inhibits the conversion of DHF to THF. So these are the anti-metabolites which target the folic acid metabolism. Cycloplasmic membrane structural function, polymyxin and daptomycin, they inhibit this plasma membrane. We have lipid biosynthesis, which is inhibited by the plantamycin. Then we have this on this RNA and DNA. So first is the DNA guidance, the inhibition, which are done by quinolone, nalidixic acid, ciprofloxacin, and novobiosine. RNA elongation is inhibited by the actinomycin. DNA-directed RNA polymerase, it is inhibited by the rifampicin, streptovaricins. Protein synthesis, 50S, these are two types, 50S and 30S inhibitors. 50S inhibitors are the erythromycin, chloramphenicol, clindamycin, linearomycin. And protein synthesis inhibitors, 30S are the tetracycline, streptomycin, kanamycin, amikacin, and nitrofuran. And tRNA inhibitors are the mupirocin and puromycin. So these are all the mechanism of actions which are given in a single figure. And I find this is very, easier to remember in this way for in case of this antibiotics. I know there are very much to remember, but nothing can be done. You have to read and you have to remember. Okay. So this was the last topic or last thing that I have to teach today. So we have completed successfully our 12-week course of medicinal chemistry and also along with the summary session. Although today it is the last class, please feel free to reach me if you have any doubt or any query regarding any question. I will try to answer your queries via email. Okay, this is my email ID. So it was our last class for this medicinal chemistry course. I thank you all for joining and answering throughout the class. I hope the classes were useful for you and you have understood a little bit of this course of medicinal chemistry better. So I will upload this PPT and this video after today's class. Okay, you can go through. All the materials are uploaded on the SWAM portal. Just you have to go to the particular link and you have to download the PPTs and you can read from there. Okay, and please follow the book, The Medicinal Introduction of Medicinal Chemistry by Graham L. Patrick. The book is very good for your course and that will be very helpful for you. So yeah, thank you. So I hope I could help you for throughout the your course. Okay. Please let me know if you have any doubt or query because it is the last class. I will not take any class after that. But the email ID is given to you. I think all of you have my email ID. So if you have any doubt or any query, please let me know through the email. Okay. All the best for your examinations. If there is no query, I will close the session for today. Thank you all for interacting. Thank you all for joining and interacting so well. Okay. So I am closing the session. Thank you all once again. I will upload the course, all the materials by today itself. Please go through them and all the best for your examination. Okay, so where you can find the PPT, you have to go to the SWAM portal. Okay, I think you all know, have you all have the access on this SWAM portal on the announcement page where the weekly announcement are there. On the left panel, you will find at the end there is a problem solving session January 2026. Clicking on that, you will find there is a problem solving session recording. If you click on that part, you will have to click on this page. Okay, if you click on this, you will be redirected to a page which looks like this. All date wise, it has all the recording for each and every class, and also there is a presentation folder link. If you click on this presentation folder, you will be redirected to a Google Drive folder which looks like this. It has all the PPTs. Okay, and all this Google Drive link is open. All the PPTs are there. You can download the PPTs easily from this Google Drive and you can read. Okay, and for the videos, you can go through all the YouTube videos, whatever it has been posted. Thank you all. All the best for your examinations. I will close the session for today if there is no query. It was a very good teaching all of you the medicinal chemistry course and all the best for your examinations and for your future. Okay. I am closing the session for today. Thank you.